Welcome to the unit on cells. What we want to take a look at is how all of those different macromolecules combine together to actually form cellular structures. The cell is the simplest structure of life. Life doesn't exist smaller than a cellular level. Now, plenty of organisms exist and live based upon lots of cells, but we do have things out there that are considered single cellular, or you'll hear the term unicellular. There's actually more life on Earth that is unicellular or single-celled than multi-celled. So when we explore cells, we want to take a look at the cell theory. Now, the cell theory says all organisms are composed of one or more cells. The cell is the basic unit of organization, and then it builds from there if it's multicellular. And new cells will arise from previously existing cells. So as we explore the cell theory, we're going to take a look at the differences between cells, cell types. We'll talk about single-celled organisms as well as multi-celled organisms and see what are the variations out there. So as biologists, we want to explore cells to understand the basic fundamental unit of life. Now, when we look at the cell sizes are limited. Gigantic animals like whales do not have gigantic cells. They just have a lot of cells. But what we see is a direct relationship. As the size of the cell increases, the volume increases. And there's going to be a maximum volume that a cell can hold before the cell is no longer functional and structurally able to work. So think about water balloons. As you fill a balloon up with water, there is an optimal size where it's perfect, it fits, it works well, and it's a great water balloon. If you continue to fill that with water, then the balloon gets bigger, it gets unwieldy, it doesn't work well, and it becomes non-functional, and sometimes it just pops. Cells are the same way. So all cells have an optimal size that they're trying to reach, an optimal volume, and they never get beyond that. If they are put into different environmental conditions in which it causes the cell to increase in size, cells can actually rupture. And we'll talk more about that when we get into diffusion and osmosis. If you put cells into certain solutions, you can actually cause them to swell up and explode. So an example we'll discuss in the future, red blood cells. The human red blood cell, if placed in pure distilled water, it will swell up and pop like an overinflated water balloon. So we always try to keep cells in their optimal range for environmental conditions so they stay in their optimal size. So again, it's limited, size is limited, and as the surface or the size increases, the volume increases, surface area increases, but not proportionally, and that limits the exchange of nutrients, and it creates problems for the cells. So we want to again, keep that in mind as we explore the cells. Now, all cells, whether you're single-celled or multi-celled, all the cells are going to share certain common features. So we want to fill in the blanks here. Key features. They all have a region of genetic material, DNA. Now, depending upon the cell type, the DNA may be housed inside of a nucleus or just kind of scattered within the cell, within the central region. All cells have cytoplasm. This is going to be the fluid part of the cell. It contains fluid, cushions the organelles, acts as a medium within the cell. All cells have ribosomes that assist in building proteins, and proteins are going to be an essential structure for cells. We talked a little bit about proteins earlier. We'll be getting into a more deeper conversation as we get into the cell membrane chapter. In all cells, no matter what you are, every single cell on the planet is surrounded by a plasma membrane. That is a membrane that is made up of phospholipids. It's a bilayer. It gives the cell its basic membrane structure and shape. Some cells have additional surrounding membranes or walls or structures, but at minimum, they all have a cell membrane, or we call it the plasma membrane. Okay, so the first group of cells we want to explore are prokaryotic cells. these are going to be known as the bacteria. But we also want to keep in mind the archaea. Archaea are considered prokaryotes, but they're different than the bacteria. Our main focus, though, is going to be the bacterial cells, looking at their shapes. So the underlined bacterial shapes wording takes you out to a YouTube video that shows some pretty cool bacterial cells. About 2.5-minute video, 3-minute video. Take a quick look, come back into the lecture, check out the rest of the stuff on the prokaryotic cells. But in general, prokaryotic cells are small, single-celled. They tend to be a bit simpler. Not as many structures inside and as many different organelles. They do have a plasma membrane, which we can see here, that inner layer. They have, in this case, they have a cell wall. They have another layer called a capsule. So the cell wall gives it shape, structure, rigidity. The capsule often gives it additional protection and rigidity and structure. There will be all these little dots inside called ribosomes. Those are the structures built for protein synthesis. They're protein factories is what they are. And we see the cytoplasm. That's that fluid area. Now the genetic material of bacteria, in this example, is this big squiggly, elongated pile of string called a nucleoid, or we refer to it as a nucleoid region. That is the DNA of that bacterial cell, of that particular prokaryotic cell there. And a big feature when we look at bacterial cells and prokaryotes is that they lack these membrane-bound organelles. We will see those when we get into the eukaryotic cells. So prokaryotic is a grouping of cells. It's not a kingdom, it's not a domain, it's not a classification level. It's just kind of a grouping of cells that we put the archaea and the bacteria domains within. So again, do not use it as classification, just kind of a grouping or organizational scheme. Alright, so within bacteria, what we will see are 3 general shapes. You have your spherical, which are round, or known as cocci. You have your rod, which look like these little pills. That's the label B there. Those are known as bacillus. And then you have the spiral. Looks kind of like a corkscrew or a squiggle. Those are also referred to as spirillii, so squiggly ones. So when we deal with bacteria, whether you go to the doctor or you're studying under a microscope, shape helps us categorize bacterial cells. Once we identify shape, we then need to figure out what makes up this particular bacterial cell, particularly what makes up the cell wall. Because if we're dealing with a pathogenic bacteria, and it's a spherical one, or coccus, we need to know how to combat that. And different antibiotics work against different bacteria. This is why please never share antibiotics with friends. If you're taking a prescription and your friend gets sick and you hand some of your antibiotic to them, it may actually feed the bacteria that your friend has instead of killing it. So to understand how to kill the bacteria or eliminate it, if it is a pathogenic or harmful one, we need to know what kind of defense mechanisms that bacterial cell has. So we start with shape. Shape helps us categorize. Do we have a spherical, a rod, or spirilla? And then we look at the cell wall. And in bacteria, a key feature of domain bacteria is that their cell walls have this polysaccharide called peptidoglycan. All bacteria have it. They have a certain amount. Thick, big, large layers of peptidoglycan are found in what we call gram-positive bacteria. They tend to stain dark purple, dark blue color. Bacterial cells that have a thin layer of peptidoglycan, those guys stain red, kind of more of a reddish, maroonish color, and those are called gram-negative bacteria. Now, the peptidoglycan helps support the cell wall, does a lot of different functions. But if this is a bad bacteria we're trying to get rid of, we need to know which type of defense mechanism they have for their cell wall. Is it a big layer or a small layer of peptidoglycan? Once you identify that, then we can try to destroy it by using an antibiotic. And so a thick layer is gram-positive, thin layer is gram-negative. Gram-positive, that purple or dark color, gram-negative, that red, maroon type of color. Okay, so those guys are out there. We'll explore bacteria a lot more when we go into Bio 112. We'll spend more time working on that. But think about it this fall. If you go to the doctor and you have a cold or a flu, and they identify it as a bacterial infection versus viral, flus tend to be viral, but they may say, no, it's not the flu, it's actually a bacterial infection. They'll probably say you have pneumococcus. Pneumo refers to the type, coccus refers to the shape of the bacteria, or if somebody's ever had streptococcus, strep throat. Cocci or coccus refers to the shape of the bacteria. Okay, so that's our bacteria domain, or the prokaryotes we would put into the bacterial group. We'll briefly just mention the archaea. Archaea, single-celled, fairly simple. We don't know much about all the archaea yet. We're still learning. This is actually a great area of research if anybody's interested in trying to do something new in research and science. But archaea, when we look at their cell wall, it lacks peptidoglycan. That's one of the big defining features that help us identify if it is archaea versus bacteria. Lack peptidoglycan, and most archaea are found in extreme environments. So the image to your left here, this is what's called a black smoker in the bottom of the ocean. The ocean's crust cracked, and magma is seeping up into the ocean water, and that looks like black clouds. That is actually smoke and methane gas being produced from the magma, the liquid molten magma combining with the salt water. It creates an incredibly, horribly insane environment, yet archaea live there. You go down and you sample that environment, there is life there. So the archaea are surviving off of the methane. That's their food source. They don't need sunlight. The picture to the right is actually a hot spring. You step in that, you boil to death. Superheated water comes to the surface in different parts of the world. Yellowstone National Park, the Old Faithful geyser, has hot springs in it. You go in it, we would die, but there's archaea living in there. Scientists are really just starting to understand these things, and there's a whole lot that can be done. There's even research being done on the inside of volcanic craters, looking at the bacteria that live in there. Nobody thought life could exist inside of a volcano, but we're finding archaea living there. So again, it's a field that still has a lot of room for exploration. Huge opportunities exist here for scientists eager to do something new and novel. But in general, archaea, cell wall that lacks peptidoglycan, and they're found in extreme environments. Right now we kind of group archaea into three groupings. We call them methanogens, they use methane. We call them halophiles if they like salty environments, and we call them thermophiles if they like hot environments. So those are your archaea and your bacteria. So in the next part of the lecture, we'll take a look at and talk about the domain eukarya and what we call the eukaryotic cells and how those cells work.